a hollow parasite that still has leaves and stems, but its internodes are extremely long and slender and its leaves are tiny. It is producing adventitious roots that develop as haustoria, penetrating to the vascular tissues of the host, justitia. Figure B22b Prosopanch americana is holoparasitic, and most of its highly modified body remains underground. It has no need for photosynthesis, obtaining all its water and nutrients from the roots of its host. This flower is also highly modified. The black and white structure is a massive set of fused stamens. Figure B22c The small cluster of cells with large red stained nuclei are part of the body of Tristerix aphyllus, a holoparasite whose vegetative body grows entirely within the body of its host, in this case, the cactus Trichocereus chilensis. The body of T. aphyllus consists of just parenchyma and lax leaves, stems, epidermis, and virtually all other features of an angiosperm body. Figure B22d The dark blobs do not belong to the visible shoot. Instead, they are the flowers of a holoparasitic plant, Pilostyles thurberi, growing completely inside its host, the shrub Dahlia. Holoparasites typically must emerge from underground or from inside their hosts when they flower. Figure 221 Simple diagram representing the raw materials and products of photosynthesis, carbohydrates produced by photosynthesis are subsequently converted to many other compounds. Respiration is another type of energy metabolism and it occurs in all organisms, with no exceptions whatsoever. As just mentioned, glucose is used to store energy and to move it from organ to organ. But a problem with glucose is that it stores too much energy for certain needs. Cells force many metabolic pathways to run by adding a small bit of energy to 2 or 3 steps of the reaction. A molecule called ATP provides just the right amount of energy for most of the steps. Glucose has about 36 times as much energy as is carried by ATP. Respiration breaks down glucose and transfers its energy to ATP. During photosynthesis, light energy is captured as carbon dioxide combines with water, producing oxygen and glucose. Respiration is more or less the reverse. Oxygen and glucose react, producing carbon dioxide and water with the energy being transferred to ATP rather than being given off as light. Plants also need minerals such as nitrogen, phosphorus, potassium, iron, and others. Plants use these in much the same way we do. Phosphate is needed for ATP, nitrogen becomes part of amino acids and nucleic acids, iron is attached to molecules that transfer electrons, and so on. Also like us, plants are not especially effective at recycling minerals. We lose minerals as we sweat, urinate, and defecate. Plants lose minerals as they shed their leaves, withered flowers, and fruit. They pack their seeds with minerals that the seedlings will need, and all those minerals are provided by the parent plant. The amount of minerals a plant needs is surprisingly small. The ash that remains after a fire contains all the minerals that had been present in the wood. Figure 222 Only the leaves of these palms are green and photosynthetic. They are the only parts of the trees that produce sugar. All other parts, such as the long stems and the underground root systems, have no chlorophyll and must obtain all their energy in the form of sugars transported downward from the leaves by phloem. Plants are master chemists. Starting with just glucose, minerals, and water, they synthesize every single molecule they need for all aspects of their life. They synthesize all amino acids, nucleic acid, and fatty acids. They make all the pigments of flowers and fruits. They make waxes, oils, resins, and fragrances. And poisons. Many plants defend themselves from pests with an amazing array of poisons. Some are bitter or irritating, others kill animals quickly. Some of the most toxic chemicals interfere with cells in the brain and nervous system. Although we humans need a complex diet that includes several amino acids, fatty acids, and vitamins that we cannot make for ourselves, that is never true of plants. Their balanced diet is sunlight, carbon dioxide, water, and minerals. Figure 223 Adenosine triphosphate. Breaking off the last phosphate to produce adenosine diphosphate results in a more stable set of electron orbitals, and energy is given off. The same is true of removing the second phosphate, but not the third. Plants and people. Box 2 to 3 toxic plants. Consider two facts: plants live among herbivores, and the world is full of uneaten plants. What is the cause? Many plants produce toxic compounds. Poison ivy, death camas, poison hemlock, and deadly nightshade are familiar examples, but the full list of toxic plants is very long. Many plants we think of as being non-poisonous are only harmless because we stop eating things that are bitter or that burn our mouths. Few people would eat enough chili peppers to get a lethal dose. The following are some toxins in plants we live with. Fascinating and well-illustrated accounts of many are given in Handbook of Poisonous and Injurious Plants by Lewis S. Nelson, Richard D. Shea, and Michael J. Ballack. Figure B2-3. Each leaf of poison ivy has 3 leaflets and the defensive compound urushiol. Anticholinergic poisons. Many nerve cells have receptors that detect the presence of a small molecule called acetylcholine, which causes a reaction in the nerve cells when it binds to the receptors. Plants such as Atropa and Datura produce chemicals that bind to one of these receptors, preventing acetylcholine from stimulating proper nerve transmission. Nicotine-like alkaloids. Alkaloids are small molecules that contain nitrogen and somewhat resemble amino acids. Alkaloids of conium, Nicotiana, and sophora block another acetylcholine receptor. Symptoms occur in the parasympathetic and sympathetic parts of the nervous system, the brain, and the junctions where nerves attach to muscles. Convulsant poisons. The small tree strychnus produces strychnine, which causes motor nerve cells to be hyperexcitable and results in convulsions. Several other plants produce convulsants. Capsaicin. Chili peppers such as habaneros, jalapenos, and tabascos in the genus Capsicum contain capsaicin, which induces some of our sensory nerve cells to release a chemical called substance P, which specifically stimulates other nerve cells that normally respond when we are being burned. Artificial selection by humans has resulted in chili peppers that range from having almost no capsaicin to some that are dangerous. Capsaicin is hydrophobic, and thus, drinking water will not cool your mouth. However, the fat in cheese, butter, and milk will absorb it and give some relief. Cardioactive compounds. Digitoxin and digoxin in digitalis inhibit a set of proteins that use the power of ATP to pump sodium and potassium across a membrane. When this pump is blocked in heart cells, it leads to an increase in the concentration of calcium within the cells. Although foxglove leaves are injurious if eaten improperly, these chemicals are used medicinally to slow and strengthen heartbeat. Cyanogenic glycosides. These are non-toxic in the plant, but enzymes in our digestive system cleave them into 2 parts, a sugar and cyanide. Cyanide inhibits the final step in aerobic respiration, preventing generation of ATP. These toxins are found in some of our most common fruits, malice and prunus, but in parts that we do not eat such as apple seeds or peach pits. Poisoning most often occurs when apricot seeds are used as components of herbal medicines. Mitotic inhibitors. Microtubules create the spindle that pulls chromosomes to opposite ends of the cell during nuclear division. The alkaloid colchicine in autumn crocus, Colchicum autumnale, prevents the assembly of microtubules. Cells exposed to this poison cannot form a spindle, and division fails. We humans have many tissues with rapidly dividing cells, such as the lining of our digestive tract, bone marrow cells that produce red blood cells, and seminiferous tubules in men. Catharanthus roseus produces the alkaloids vinblastine and vincristine that also block cell division. These are used in chemotherapy to stop cancer cells from dividing. Toxalbumins. These proteins bind to ribosomes and prevent them from translating messenger RNA into protein. Ricinus produces the toxalbumin ricin, one of the most toxic substances known. Jatropha species has a higher concentration of a related toxalbumin, and a single seed can cause severe poisoning. Urushiol. About half of all people are allergic to urushiol, an oily substance in species of Toxicodendron. All parts of these plants can trigger an allergic reaction. Remember that these plants do not have toxic compounds in order to protect themselves, or with the purpose of keeping herbivores away. It is just that toxic or injurious plants are more likely to be left uneaten compared with non-toxic plants and therefore are more likely to produce more seeds and more progeny. Table 2-3 Chemicals that plants produce starting with glucose. Overview of information in plants. A universal feature of all known life on Earth is that DNA is used to store information. Just as with energy and matter, organisms take in information, store it, move it from place to place, transform it, use it, and then pass it on to their offspring when they reproduce. The greatest source of information for any individual is its parents, the DNA contained in the sperm cell and egg cell that initiated the life of the individual. In plants, DNA is stored in the nucleus of each cell, as well as in plastids and mitochondria, which are also present in all living plant cells. When a cell needs to use some of the information, it transforms the needed information by making messenger RNA, and then it transfers that information to the area of the cell where it is needed. Most of the information is further transformed as the messenger RNA guides the production of specific proteins. Smaller pieces of RNA, often called microRNA, act by binding to various components of the cell, activating some, inhibiting others, and in general fine-tuning the activities of many genes. The DNA of each species is unique. For example, cherries and peaches have almost identical DNA, but the information in several genes in one differs just enough from that of the other so that cherries are small, red, and shiny whereas peaches are large, yellow, and fuzzy, and each has distinct flavors and aromas. A plant, just like any other organism, also uses information from the environment. Environmental information must be perceived then transferred to where it is needed, and finally the plant must respond appropriately. While the plant is still nothing more than a dormant seed, the environmental information it responds to is often very simple. It is warm enough to germinate. As temperatures climb from winter to springtime, at some point they are warm enough and many seeds then respond by becoming active, respiring more rapidly, and germinating. Seeds of other species remain dormant because they are dry, not cold. They need to detect when their surroundings have become wet enough to support a growing seedling. The environment also supplies a large amount of more complex information, in many forms. Sunlight provides the energy for photosynthesis, of course, and it also indicates the direction up from down. In all parts of the world except near the equator, sunlight provides information about seasons. Days are shorter in winter, longer in summer. By detecting this, a plant measures the season and responds by flowering and fruiting at the correct time and by becoming dormant in autumn. Roots are not exposed to sunlight, but they detect gravity and thus know which direction is down. Deeply buried bulbs also detect gravity and respond in two opposite ways. Roots follow gravity downward, whereas shoots grow upward. We are now learning that some plants are much more perceptive than we ever suspected. Tendrils bend toward anything that gives them even the slightest touch, leaves detect the growth of a single fungus spore on their surface, and some orchids detect whether a pollinator has merely landed on one of their flowers or if it has actually taken the pollen. Figure 224. All plants here obtain energy from their environment, but think of the two trees. They both obtain information about which way is up. For the grasses in the pasture, up is in the direction of the brightest light, but for the tree on the right, the brightest light is away from the larger tree that is shading it, so bright and up are not the same thing for it. These trees also need to detect the approach of winter and upsize their leaves while they can, and they probably detect the presence of many insects and fungi as well. Figure 225. When these bulbs became active and started growing, all parts were so deeply buried in soil that they could not detect any sunlight. But both new roots and new leaves detect gravity, with roots growing downward, leaves growing upward. Various parts of a plant itself create information that informs the rest of the plant of their activities. Healthy leaves produce hormones that are carried to the roots. A large amount of hormone indicates the leaves are active and need water. The roots respond by growing and absorbing water and nutrients. In autumn, as leaves prepare to be shed, they stop producing the hormone, so the amount of the hormone decreases, indicating the leaves no longer need water. Roots respond by slowing their growth and conserving their energy. After a flower is pollinated, the developing fruit sends signals indicating that it needs nutrients for itself and the embryonic seeds inside. This communication among the parts of a plant is necessary to keep the plant integrated and all parts developing in coordination. It is especially remarkable considering the long distances involved between the thousands of leaves, flowers, branches, and roots of a large tree. Overview of plant diversity and evolution. All the thousands of species of plants of the world are not just a hodgepodge of randomly associated features. Instead, there are several large, recognizable groups such as mosses, ferns, conifers, and angiosperms. Within each group, we are likely to find certain features and unlikely to find others. For example, mosses never have xylem or phloem and they never have seeds either. As a second example, ferns always have vascular tissue but they never reproduce with seeds. Several aspects of the groups in Table 2.4 are not quite accurate, but the groups and terms presented there have been used for more than 100 years, and you will still encounter them often in books about gardening and roadside flowers. An overview of them will be given here. Definitions of important terms are grouped together in Table 2.5. Mosses, liverworts, and hornworts are often grouped together and called bryophytes. They are similar to each other because they not only lack vascular tissues and seeds, but also because plants in all three groups are haploid. Mosses, liverworts, and hornworts are unique among plants because they live their lives with only one set of chromosomes, not two. Their bodies are very simple, not much more complex than the algae that were the ancestors of plants. Table 2.4 Overview of the main clades of organisms. Table 2.5 Terms used for plant groups. Names in formal classifications are capitalized but informal names are not. The informal names of some of the groups here are angiosperms, conifers, cnidophytes, and mosses. Their formal names are angiospermophyta, coniferophyta, cnitales, and mussae or bryophyta. All other groups of plants have vascular tissue and are diploid. Many vascular plants resemble bryophytes in being unable to form seeds. They reproduce using spores and are typically called the spore-bearing plants. These are often referred to as ferns and the fern. Allies. An old way of thinking about plants is that ferns and fern allies had advanced above the bryophytes as they evolved to have vascular tissue, but then their evolution stagnated, while other vascular plants continued to advance evolutionarily to the point where they could make seeds as well as vascular tissues. The more advanced seed plants then divided into two groups, those with naked seeds and those with clothed seeds. Here, clothed seeds refers to seeds formed inside a fruit in contrast to naked seeds that are produced in a cone. The term naked refers to the time when a pine cone is extremely young. At that point, its scales are far enough apart that you can look in and see the individual naked ovules. The carpels of angiosperms are always sealed, and the ovules are never visible without cutting the young fruit open. There are other differences between gymnosperms and angiosperms that make us think of angiosperms as being more advanced. Angiosperms have more complex wood and leaves, as well as several modifications that improve photosynthesis. Angiosperms also cooperate with animals that pollinate their flowers and distribute their seeds, whereas conifers never rely on animals for pollination, and only a few cooperate with animals for seed dispersal. Figure 226 Mosses cover the rocks in this stream in the Olympic National Park. Mosses have no xylem or phloem and cannot conduct water very well, so they are typically very small plants. Although we associate mosses with damp places, many are adapted to deserts, being inactive except on rare days when there is rain, fog, or dew. Figure 227 These are plants of the leafy liverwort Bassania trilobata. As with mosses, the bodies of leafy liverworts consist of a stem and leaves, but never any roots, xylem, or phloem. It is usually necessary to have a hand lens or magnifying glass to distinguish between a leafy liverwort and a moss. Figure 228 This is a rather common thalloid liverwort that is easy to recognize. Its bodies never have any hint of leaves or stems. They are flat and about 1 centimeter wide, and they occasionally fork into two growing points. Their upper surface has a distinctive diamond-like pattern that resembles reptile skin. Figure 229 hornworts such as this Phaeoceras laevis are widespread, but they only thrive when it has been rainy for a while. They grow in shady places, sometimes on bare soil, sometimes mixed among grasses. Each long projecting cylinder is the horn and is actually the diploid portion of the plant. Each horn is the same phase of the life cycle as is a tree, bush, shrub, or any other familiar plant. The small flat patches of tissue in contact with the soil are the gametophytes, the haploid part of each plant. It is the part that is similar to mosses, leafy liverworts, and thalloid liverworts. The life cycle of plants is much more complex than that of animals, but is fascinating. We can arrange the different groups in a linear sequence that seems to reflect plant evolution. The earliest true plants were more advanced than algae because they had simple tissues and organs but little else. This would be the moss level of evolution. Other plants advanced to the level of having vascular tissues, others continued evolving to the point of having seeds. Then one group evolved more than all the rest, to the point of having flowers and fruits. Some would even refer to angiosperms as the pinnacle of plant evolution. In this method, emphasis is placed on the level to which a group has evolved. The technical word for level is grade. We are grouping plants into grades, with the lowest grade being non-vascular plants, the highest grade being angiosperms. We still encounter this concept of grade when people refer to the lower plants and higher plants. This grouping by grade is very handy if you are not too worried about accuracy, but we now have a newer method of grouping plants, one based on groups that consist of all the species that evolved from a common ancestor. Such a group is a clade. Fortunately, grouping plants into clades gives us almost the same result as grouping them into grades. We need to abandon only a few of the old terms. For example, we no longer use the term gymnosperm. We suspect, but are not at all certain, that seeds evolved only once. If so, then all seed plants alive today descended from that original species of seed plant, and we say it was the common ancestor to all seed plants. Some of its descendants evolved into cycads, others became ginkgos, another group evolved into conifers, and still another group became angiosperms. There are two important points here. The first is that a clade consists of an ancestor and all its descendants, so the group, seed plants, is acceptable, it is a clade. But the word gymnosperms refers to all seed plants except angiosperms. It includes the common ancestor of seed plants but only some, not all descendants, so gymnosperms is not a clade. We prefer not to use the term gymnosperm if we care about being accurate. Figure 230. This Selaginella lepidophylla is used as an example of a fern ally because of its interesting biology. It is a resurrection plant. It is a diploid vascular plant with xylem and phloem, and it grows in harsh deserts with long dry periods. This photograph shows the plant after autumn rains, when the plant is hydrated, healthy, and active. Compare with figure 230b. This is also Selaginella lepidophylla, but after a long dry period, the plant has curled up, minimizing the amount of surface exposed to dry air and UV light. Although it looks dead, it is not only alive but very healthy, merely dormant, somewhat like a dry seed that is alive. After a rain, these plants would uncurl and look like those in figure 230a if forcibly uncurled now. The inner parts would be bright green. The second important point is that the ancestors of any clade of living organism all lived in the past, and for clades such as angiosperms or conifers or seed plants, the ancestors lived in the very distant past. Consequently, we can never be completely certain which ancient, extinct group was the common ancestor of any modern clade. Instead, we have to study the living organisms and try to infer the features the common ancestor had. Using angiosperms as an example, all modern as well as fossil angiosperms have vascular tissues. They produce flowers and they make seeds. They never make anything similar to a pine cone, and they are never haploid like bryophytes. From this we assume that the common ancestor of all flowering plants was a diploid vascular seed plant that made flowers but not cones. At present, we are able to analyze the sequence of nucleotides in the genes of organisms, and at present we have sequenced all the genes of many species. This provides an abundance of new information for constructing and testing our theories and models. Figure 231. These plants of Equisetum telmatea are diploid vascular plants that are now considered to be part of the ferns. These are descendants of an ancient group. Fossils of plants that have similar features can be found in rocks as much as 150 million years old. Equisetums are called horsetails or scouring rushes, and are distinctive in having a very coarse surface. They can be found almost everywhere, along streams and along railroad tracks in moist areas. Figure 232. This is a single plant of sword fern. It has a short vertical stem that supports the many leaves, and below ground are many fibrous roots. Ferns are diploid and have xylem and phloem, but their spores grow into tiny haploid plants that resemble the bases of hornworts. This large sword fern pictured here is the equivalent of a hornwort horn. In rainy areas of the Pacific coast of the United States, sword ferns are so abundant they resemble meadows of grass. Indeed, millions of years ago, before grasses evolved, most grasslands were actually huge patches of ferns. Ferns and dinosaurs were abundant at the same time. Long before grasslands existed, the herbivorous dinosaurs ate ferns and fern allies. Figure 233. This cladogram is a hypothesis about the ways in which modern plants are related to each other. At the very base would be the ancestral plants, not much different from algae. Many years later, at B, some plants began to evolve into liverworts while the rest of the plants continued evolving other features. At C, some plants began to evolve into mosses, and so on. At some point between D and E, vascular tissue evolved in the main group of plants, and all plants that descended from these early vascular plants still have vascular tissue as well. That means that the xylem in ferns resembles the xylem in flowering plants not by accident but because both evolved from the same ancestral xylem. The lines that show the ancestry of cycads, neophytes, ginkgo, and conifers all start from the same point. That means we do not yet know which diverged earliest, and we do not know which group is most closely related to which other group. The line for the ancestry of angiosperms diverges a bit later, but it could be that this is incorrect and we should draw it coming from G, the same point as the cycads and others. The old concept of grades assumed that cycads, ginkgos, and conifers were evolving more slowly than angiosperms and, given enough time, they would finally at long last evolve up to the level of having flowers. Similarly, maybe one day in the future, ferns would evolve to the point of having seeds, and so on. It is like moving from grade to grade in school. Everyone starts out in the first grade and then all move along toward graduation, some moving faster than others, some stopping completely. This is the second problem with grouping plants into grades. It assumes that all plants have the same goal, that having vascular tissue and seeds and flowers is the optimum way a plant can be adapted to its environment. We no longer believe this. We now realize that there are multiple ways for an organism to be adapted to its environment. This is reflected in the questions, what are the alternatives, and what are the consequences? Consider mosses and angiosperms, two groups at the opposite end of the old grade system. Mosses are tiny and simple whereas most angiosperms are large and complex. But mosses are very well adapted to their environment. They thrive in low light, on brick walls or bare stone. They need only enough sunlight, water, and mineral nutrients to make a few spores and then they can reproduce. In the moss clade, evolution by natural selection has found solutions to survival that work well for small, simple plants. Having vascular tissue and seeds and so on would not make a moss any more adapted than it is without them. This is a crucial point I will emphasize many times. There are many, diverse ways to be adapted, numerous ways of thriving. There is not one single structure or physiology that will be optimum for every organism. No organism is a pinnacle of evolution. Botanists are working harder than ever trying to figure out how plants are related, which clades are valid. The following are the ones you should know as you continue into this book. Plants are a clade. True plants originated from green algae only once. The technical name for plants, to distinguish them from algae, is embryophytes, but this is used in only the most technical writing. It had been suspected that mosses, liverworts, and hornworts might have had a completely separate origin. Perhaps they did not descend from the same ancestor that produced all the other plants. But the sequences of nucleotides in the genes of bryophytes are so similar to those of all other plants that we are now confident they too are true plants. The terms vascular cryptogams and fern allies are handy but not accurate. Early diploid plants diverged into two clades, both with vascular tissue and without seeds. One clade, which we call the lycophyte clade, was very successful initially and evolved to be large trees with many complex features, but almost all became extinct. At present, the surviving descendants are Lycopodium, Selaginella, and a few other genera. These are the fern allies you will see in wildflower books. The second clade was a bit slower at the start, and for millions of years was overshadowed by the abundant members of the lycophyte clade. Many of the early members became extinct, but this clade produced all the ferns and seed plants. The term most often used now is the euphylophyte clade. The ferns make up a subclade of the euphylophytes, and they have been very successful without having seeds. Ferns thrive in all sorts of habitats, not only shady, damp forests, but also hot, dry deserts. They never produce wood, so they are all herbs, even the very tall ferns called tree ferns. Some ferns have evolved to have sophisticated relationships with ants, relationships that are mutually beneficial. The ferns make up a well-defined subclade within the euphylophyte clade, but we are not certain how to group the remaining vascular plants. Many are known only as fossils of isolated leaves or wood or reproductive organs. It is very rare to ever find a fossil of an entire plant with all parts still attached to each other. Some especially intriguing fossils look like fern leaves with seeds on them, so they were named seed ferns. But no seed fern is alive today, and no living fern ever has anything that even slightly resembles a seed. We believe that the name seed fern was a poor choice, but by now it is very commonly used. The many fossils of seed ferns are too incomplete for us to know if there was only one group of early seed plants or several. If there was only one group of seed ferns, then probably seeds evolved just once. If so, all seed plants make up one clade, the seed plants. But if seeds evolved 2 or 3 times, then seed plants is not a clade, not a closely related group that all descended from the same common ancestor. If seeds evolved several times, seed plants would then be the name of a grade and could only be used informally. Other extinct seed plants lived after seed ferns. Many look like intermediates in the evolution of modern seed plants from seed ferns, but the picture is not yet clear. Many people are taking a cautious approach now, tentatively considering all the seed plants as being related. They assume that seed plants evolved only once, and then diverged into several major groups, most of which have some members that are still alive today. According to this theory, the early seed plants gave rise to these clades: cycads, neophytes, ginkgo, conifers, and angiosperms. Until recently, it was thought that all members of the angiosperm clade were either monocots or dicots. We used to think that after the first flowering plants originated, the group immediately diverged into 2 distinct clades, monocots and dicots. But new evidence indicates that split did not occur immediately. Early flowering plants evolved and diverged into several groups before the monocots originated and became so distinctive. The term dicot had referred to all the angiosperms except monocots, but like the term gymnosperm, this is unacceptable because it does not include all the descendants of the common ancestor of the group. At present, we use the terms basal angiosperms for those angiosperms that diverged early, monocots for the clade of lilies, palms, agaves, orchids, and their relatives, and eudicots for all the rest of the angiosperms. You will still see the term dicot used in many books, and it most often means basal angiosperms plus eudicots. Although the terms dicot, gymnosperm, and fern allies are being abandoned, it is good for you to know why they were invented and how they were being used. Even though you will still see them occasionally, it is best if you avoid using them. Figure 234. These avalanche lilies are shown as representative monocots, but the monocots are a large, diverse group. Many have large showy flowers, although flowers of grasses and sedges are small. Most monocots have parallel venation in their leaves, and many die back to a persistent subterranean bulb or rhizome. These Joshua trees are shown as another representative type of monocot. Many people think of monocots as small, delicate plants like lilies, irises, crocuses, and so on, but many, such as Joshua trees, become very large and tough. Other strong monocots are palms, agaves, yuccas, bamboos, and many bromeliads. This lupin, Lupinus latifolius, is shown as a representative eudicot, but eudicots are a gigantic and diverse group. Of the plants you see every day, unless a plant is obviously a monocot, conifer, or fern, then it is probably a eudicot. Eudicots can be trees, shrubs, herbs, vines, parasites, and succulents, as well as many other types of plants. Botany and beyond. Box 2-4 Noah's Flood and Population Biology. The biblical story of Noah's flood in Genesis provided a powerful stimulus for developing the sciences of population biology, biogeography, and evolution. Various biblical scholars had studied Old Testament texts, the Jewish Torah, and archaeological evidence, trying to establish dates such as the birth of Jesus and the construction of Solomon's temple. Although there is no archaeological evidence, various Bible scholars concluded that the flood of Noah occurred in 2348 BC. Noah's flood was of special interest to early scientists because at that point, according to the Bible, all life on earth had been destroyed by God except for the creatures in the ark. Scientists formulated the question, do plants and animals reproduce rapidly enough to repopulate a completely denuded earth in the time since Noah? Thus a scientist posing this question in 1700 AD, for example, could assume that Earth had been repopulated in 2348 plus 1700 is equal to 4048 years. First, consider the Bible passages that the scientists relied on. Genesis 7 verses 2 and 3 states that God told Noah to take 7 pairs of every kind of clean animal, only 1 pair of every kind of unclean animal, and also 7 pairs of every kind of bird. Genesis 7 verse 20 states that the flood completely covered the tops of all mountains. That means that there was no dry place anywhere that animals could use as a refuge and survive. Although we are all familiar with the rain lasting 40 days and 40 nights, the Bible states that it took a long time for the waters to drain away after the rain stopped. For the early scientists, the critical passages are Genesis 8 verse 4 and 5 that state that the ark was floating for 7 months before it settled on Mount Ararat, and it was 10 months before the tops of other mountains were visible and Noah opened the doors. Especially important was the explicit statement that God's plan succeeded. Genesis 7 verse 23 states that everything outside the ark had been killed. In the book of Genesis, God had set up the perfect experiment in population biology. All animals had been killed. And Earth had to be completely repopulated with all types of animals and birds, starting with just 1 male and 1 female of each of the unclean animals and just 6 males and 6 females of each of the clean animals. Could just 12 cattle breed rapidly enough to produce all the herds and wild cattle on Earth in 1700 AD? Could just 2 pigs be the source of all swine now present? The first step in answering this was to determine the fecundity of animals. How old do they have to be before they produce their first young? How many young do they produce in each litter? How many survive long enough to breed? And how many sets of offspring do they produce in their lifetime? Fortunately, it was about this time that wealthy landowners began keeping track of these sorts of numbers for cattle, poultry, fish, fowl, and even the deer in their private forests. The answer for every type of animal was the same: yes, all animals produce young rapidly enough to repopulate Earth in much less than 4,000 years. The early scientists then began to refine their thoughts and reformulate the question. At present we believe in a balance of nature. Plants capture carbon and energy through photosynthesis, then the plants are consumed by herbivorous animals or by pathogenic fungi and bacteria. The herbivores are eaten by carnivores. If there are too many herbivores, they damage the plants too much, and some herbivores starve, which in turn affects carnivores. An outbreak of a pathogenic fungus that damages plants will cause various animals to starve, and so on. The balance between all these factors changes from year to year, so the relative numbers of organisms also change yearly. But prior to the analysis of Noah's flood, people believed that a different kind of balance of nature was maintained by God. God had created the various carnivores, herbivores, and plants such that plants would prosper just enough to supply the herbivores with exactly the right amount of food, and likewise the number of herbivores would be perfectly correct for the numbers of carnivores in the world. The balance would be perfect, never changing, and with no waste or shortage unless God specifically causes a drought or pestilence. As early scientists studied animal fecundity, they realized that the numbers did not fit together to make a divinely balanced system. All organisms are too fertile. If the previous interpretation of Genesis were correct, God had created plants and animals with the wrong properties to fit into a system that would be perfectly balanced and unchanging to the end of time. They realized that the story of Genesis as well as their own research pointed not to a stable, harmonious, garden-like earth, but rather to one of change and competition, of some organisms increasing at the expense of others. Population biology was born and has thrived ever since. Overview of plant ecology. Plants live in a complex, dynamic, and changing world. Earth's complexity is easy to see. Some areas are mountainous with cold temperatures and thin, rocky soil, but other areas are low, warm, and moist. Volcanoes release lava and ash that are initially sterile but that gradually change into rich acidic soil. In contrast, limestone breaks down to form alkaline soil. In addition, some soils are so rich in certain minerals they are toxic and plants cannot grow on them, whereas other soils have a balance of mineral nutrients that encourages luxuriant plant growth. On mountains and in valleys, the slopes that face the equator receive more sunlight and are much warmer than the sides that face away. Earth is dynamic, energetic, for many reasons. As Earth rotates on its axis, a particular area faces the hot sun during the day but faces the extreme cold of outer space at night. Throughout a year, seasons change from spring to summer and so on, each characterized by the amount of energy available from sunlight. Earth's atmosphere and its oceans are two gigantic fluids that circulate, moving energy from place to place. We experience the circulation as cold fronts of frigid polar air that move southward, pushing warm air aside, or as storms and hurricanes that carry water and energy in such concentrated forms they destroy buildings and push over trees. Earth's hot interior is another source of energy that affects life on the planet's surface. Erupting volcanoes catch our attention, but Earth's interior heat also powers continental drift. Hot, molten rock rises along the center line of each ocean then spreads outward as newly formed seafloor. This pushes the old sea floor to the side, which in turn pushes continents from place to place. North and South America formerly were widely separated by thousands of miles, but the continental drift of each changed direction and now they are moving toward each other. Central America is the result of this collision, which is still occurring. North and South America first made contact with each other less than 2 million years ago. As they continue to push into each other, they crumple Earth's crust, pushing Central America upward and causing numerous volcanic eruptions throughout the region. As the two continents finally came into direct contact, many animals and plants could migrate from one to the other. For example, armadillos have been slowly migrating from South America into North America, and have only recently arrived in the southern United States. Unfortunately, saber-toothed tigers migrated south, and as they spread into South America, they killed off many large animals, driving many species to extinction. Cacti originated in South America and were confined to just that continent until continental drift finally brought North and South America together. Cacti could migrate north into Mexico and the deserts of the United States as animals carried their seeds. The creosote bush that gives all the deserts of the southwestern United States their distinctive aroma also came from South America. Earth is a changing world. For comparison, think about our moon. It is complex but not dynamic. The craters we see today are the same as they were thousands or millions of years ago, with the only new activity to ever occur is an occasional meteor strike. But Earth is never the same, not even from one year to the next. Mountains are lifted by volcanism or continental collision, and they are worn down by erosion. Flooding rivers carry silt that settles into rich deltas and marshlands filled with life. Fires drastically alter the numbers of plants and animals in forests and prairies that burn. As continents drift, their changing positions affect the circulation of the oceans and thus the circulation of heat and moisture. During the time North and South America were widely separated, the Pacific and Atlantic Oceans were in contact and water circulated around each continent. But once the continents collided and formed Central America, oceanic circulation was altered. Water was forced to flow northward and southward, carrying warm water toward the poles and making those areas warmer than they had been before. Plants and animals located in the far northern parts of North America and Europe experienced climatic warming. Figure 235. This cactus, Stetsonia corine, is at home in Argentina. It is one of many South American cacti. For almost any cactus you would encounter north of the Panama Canal, there are similar but unrelated species in South America. There were no cacti in North America until continental drift brought North and South America close enough together for seeds to be carried northward. Figure 236. Creosote bush is at home in the deserts of North America, but its closest relative, Laria divaricata and two other species of Laria, live in South America. The plants in South America resemble those in North America so closely that we believe that at some very recent time, a seed or two of L. divaricata was carried northward the entire distance and then thrived in the new habitat. Once in North America, the plants gradually evolved just enough that we consider them to be a new species, L. tridentata, distinct from the ancestral L. divaricata. Much earlier than that, just before plants evolved from green algae, all continents had drifted together into one gigantic continent called Pangaea. This was so enormous that its center was a desert. Storms blowing in from the oceans would rain themselves out before they could reach the center of the supercontinent. But because all land was connected, the newly evolved plants could spread easily from place to place such that by the time Pangaea broke apart, each new continent carried some plants, and no continent was left without any plants. The first splitting of Pangaea ran from east to west, creating two gigantic continents, Laurasia and Gondwana. Imagine if Pangaea had broken apart before plants evolved from algae. Plants would have originated on one but not the other. On one continent, plants would have spread and diversified, and would have evolved into many clades. But the other continent would have remained barren until at some point millions of years later, some spores or seeds would have somehow blown or drifted to it. A crucial cause of Earth being a changing world is life itself. Living organisms take up energy and nutrients from soil, water, and air and then release their waste products back. Plants take so much carbon dioxide out of the air that they have changed the atmosphere. And because carbon dioxide is a greenhouse gas, the changing levels of carbon dioxide affect the temperature of Earth's surface, the biosphere, the place where life occurs. Photosynthesis produces oxygen as a waste product that is released into the air. Before the evolution of photosynthesis, Earth had no free oxygen at all. There was no aerobic respiration nor were there any animals. The photosynthetic production of oxygen is carried out by cyanobacteria, algae, and plants. Without these organisms, animals and fungi could not exist. Similarly, as animals and fungi respire their food, they give off carbon dioxide, which of course is valuable for plants. Thus photosynthetic organisms have changed Earth such that it is a better habitat for organisms that respire, and vice versa, organisms that respire are a source of the carbon dioxide needed by photosynthetic organisms. Figure 237. For most of us, it is difficult to imagine what Earth was like before the evolution of true plants. There was no life on land at all. For billions of years, life existed as bacteria and archaeans in the oceans. Later there were fish and algae, but nothing at all on land until at the earliest, about 450 million years ago. Most of the surface was rock, and if any soil particles formed, they would have mostly been washed into the oceans because there were no plant roots to hold them in place. The area shown here is in Death Valley National Park, which does have plant and animal life, but it is widely scattered and confined to areas with at least occasional water. Plants, animals, and all other organisms interact in more immediate, direct ways as well. Interactions may involve one organism benefiting by harming the other, by both organisms helping each other, by one organism benefiting without harming the other, and by one organism harming another with no benefit to itself. Herbivores and omnivores are examples of predation. They benefit by eating plants, and this of course harms the plants, so plants need to defend themselves from animals. There is no one single optimal defense, instead many alternative types of defense have evolved, such as having spines or being poisonous. Pathogenic fungi and bacteria also prey on plants. Figure